Hello students, today we are going to see how to determine the activity of salivary amylase using the DNSA method. So this is the experiment that we are going to do today, the activity of salivary amylase or alpha amylase using the DNSA method. Now, to start at the outset, let me tell you that enzymes, they are biological catalysts which catalyze reactions within the body, but they can catalyze the similar reactions outside in your test tube, the reactions that you do in the lab. Okay. These catalysts, they are highly specific. They can exhibit different types of specificities. Those can be reaction specificity, that is, they catalyze a particular type of reaction, or it can be kinetic specificity, where they catalyze some reactions faster than the other reactions, or it can be stereospecificity, where you talk of enzymes acting on a particular stereoisomer of a pair of isomers, or leading to formation of a particular predominantly one stereoisomer of the possible stereoisomers. If you can see in this reaction, if you see in this reaction, the carbonyl group, is reduced to give you only a particular enantiomer. In this case, it will be the S form which will be formed and not the R. So you understand that the enzymes are highly specific in nature. Okay? Normal chemical reactions which you carry out in the laboratory, they are catalyzed by changes in pH, or by increasing the temperature you can increase the rate of the reaction. Other ways by which you can increase the rate of the reaction is by adding some inorganic catalysts. You have studied many catalyzed reactions even in your green chemistry paper, or by adding the acid or alkali. You have seen how hydrolysis of esters, hydrolysis of amides can be carried out in presence of acid or alkali. So general chemical reactions, they are the rate of these general chemical reactions which you do in the lab can be accelerated by doing one of these three steps. If you talk of biomolecules, molecules like carbohydrates, like proteins, like nucleic acids, they are highly sensitive to changes in temperature or pH. So reactions involving biomolecules, they have to be carried out under specific temperature and pH conditions. So in case of biomolecules or the reactions involving these biomolecules, special catalysts called enzymes, they are used. You must know that these enzymes are mostly proteins, and some of them are nucleic acids. Okay. How do these enzymes catalyze biological reactions? They catalyze biological reactions by lowering the activation energy of the reaction. You can very well understand, if the activation energy barrier for a reaction is reduced, the reaction will be accelerated. Like other inorganic catalysts, enzymes do not alter the overall reaction rate. They only help to attain the equilibrium faster. Equilibrium, fast attainment. Now, where do these reactions involving enzymes take place? The reactions involving the enzymes take place at specialized sites, which are called the active sites. Okay. If you can see in this figure that this is the rate of an uncatalyzed reaction, and this is the rate of a catalyzed reaction. You can see here the activation energy barrier has been significantly reduced in presence of a catalyst. Okay, so the reaction is faster. Here I have depicted this is a picturized view where you see this as the enzyme, and this is the active part of the catalyst, the active site, where the substrate comes and binds, the reaction takes place, and after the reaction has taken place, the product normally the enzyme does not have any affinity for, leave the enzyme surface for fresh substrate to come and bind. This is largely how any enzyme catalyzed reaction takes place. Now, enzymes, as I said, most of them are proteins. Some of them are, simple proteins. Simple proteins in the sense that they do not require any non-protein part to exhibit their catalytic activity. Whereas some other enzymes, they require a non-protein part to exhibit their catalytic activity, and that non-protein part, which is sometimes required by the enzyme to exhibit catalytic activity, are called cofactors. Okay. Now these cofactors can be mainly of two types. Either they can be inorganic metal ions, okay, metal ions like calcium, copper, zinc, or they can be organic molecules. Now these cofactors, especially the organic molecules, can bind to the enzyme in two ways. One is when they are loosely bound. Okay, molecules like vitamins, molecules like nucleotides, they are loosely bound to the enzyme surface. They are called coenzymes. These coenzymes, they come and bind to the enzyme when the catalyzed reaction has to take place. And after the catalyzed reaction is over, these coenzymes will leave the enzyme surface. Whereas prosthetic groups are organic molecules which are tightly bound to the enzyme surface. They can be compounds like nucleic acids, can be lipids, they can be carbohydrates, etc. We will study all this in detail in the theory when we come to the enzyme topic. You know that we are going to study this. In the next week, I will be going into details about all this. But for the moment, you should know that sometimes enzymes require some non-protein part to exhibit catalytic activity, and that non-protein part is called a cofactor. How do these cofactors help enzyme action? They provide certain catalyzed groups, certain functionalized groups, which the enzyme is lacking, and these functionalized groups or these catalytic groups are required by the enzyme to exhibit catalytic activity. So the role of the cofactor largely is to provide catalytic groups which are involved in the binding of the substrate and which are involved in catalysis. Okay. For any enzyme catalyzed reaction, the measurement of the enzyme reaction rates can be done by plotting what we call the progress curve. Progress curve is a graph of the amount of substrate which is transformed versus the reaction time. You can see that the rate of the reaction is linear initially, it becomes slower, and then it becomes constant. So this happens because of four possible reasons. One is the product that is formed. It may start acting, it may start retarding the reaction. That is one thing. Second thing is it may so happen that all the active sites on the enzyme surface where the reaction takes place have been saturated. Once they get saturated, there will be no increase in the rate of the reaction. Only the substrate will be required to replace the active sites, which become empty once the product leaves the active site. The second thing, the third thing is that the enzyme may become denatured. Under the reaction condition, it may so happen that your enzyme becomes denatured. Okay. And the fourth thing is possible is that the reaction has started to go in the backward direction. So these could be four reasons why the rate of the reaction, may start to retard and may become constant after some time. Okay. So this part of the graph, is the linear curve. After it slows down somewhere here and then it becomes constant here. So for any enzyme assay, for any enzyme catalyzed reactions which you do in the lab, the following factors have to be kept in mind. You have to always use a buffer so that the pH remains constant, enzyme structure, enzyme activity is largely affected by the changes in pH. When I discuss the chemistry of enzymes and the biological properties of enzymes in the theory class, I will elaborate more on this. Second is the substrate should be unlimited. You should have enough substrate so that it can saturate the active site of the enzyme. So substrate should always be unlimited. Then you should have the presence of the activator or the cofactor or the coenzyme, which are required to accelerate the enzyme activity. And then the concentration of the enzyme should be optimal. The enzyme should be optimal concentration. Then the incubation temperature. The temperature, every enzyme has a temperature which is called the optimum temperature at which the activity of the enzyme is maximum. So the enzyme catalyzed reaction, whenever you do those reactions, it is important that you know what is the optimum temperature for the enzyme and you carry out the enzyme catalyzed reaction at its optimum temperature. And then is the incubation time. We have seen in this graph that the enzyme progress curve is linear for some time and after that the rate becomes to retard and then it becomes constant. While you are doing the enzyme assays, it is important that the incubation time should be such that the enzyme is following linear kinetics. Only then you can extrapolate and you can determine the concentration of the unknowns. Okay? And finally, the termination of any enzyme catalyzed reaction can be done either by raising the temperature above the optimum temperature of the enzyme or by adding acid or alkali in excess, which will denature the enzyme and which will lead to loss of the enzyme activity. So the enzyme catalyzed reactions, you can measure in three different ways. The first way is by doing the sampling method where samples are withdrawn from the reaction under study at regular intervals of the time and they are analyzed for the disappearance of the substrate or for the appearance of the product. This is one way of measuring the activity of the enzyme. The second way is the continuous method. Continuous method में हम क्या करते हैं कि the vessel where the reaction is taking place is connected to a hyphenated technique. Continuously the change in the substrate concentration or the change in product concentration is monitored to study the progress of the reaction. But what we normally do in the undergraduate lab is the discontinuous method. What is the discontinuous method? You allow the reaction to proceed for a certain length of time, okay? And after that, you bring the reaction to a halt. Reaction को stop कर देते हैं acid डाल के या alkali डाल के ताकि enzyme activity halt हो जाए and then you measure the amount of product which is formed or amount of substrate which has reacted in a fixed period of time. That is what we normally do in the lab, and that is how we are going to do all the enzyme experiments, in our undergraduate lab. Like I told you, for protein, it is very important for any biological assay that you do a controlled assay. A control in the last experiment that we did was substrate blank, and normally in the enzyme catalyzed reaction, we take control as the enzyme blank. So your control solution will have all the reacting species except for the enzyme. So by doing a control assay, you are putting all the absorbance, because of any of these reactants to zero, so that you can only measure the absorbance of the analyte under question, which is being formed or which is being reacted in the biological assay. So, the different analytical methods which are available to study the rate of an enzyme catalyzed reaction. One is the UV visible spectrophotometry. Second is the luminescence method. Third is the radioisotopic method. Fourth is the microcalorimetric method. And 5 is the fluorimetric method. But what we do in the undergraduate lab is the UV visible spectrophotometry. Okay. Now, measuring the enzyme activity can be done in terms of SI unit, that is called cation, represented as KAT, which is the amount of enzyme which is required to convert 1 mole of the substrate, per unit time into the product. However, for all practical purposes in the undergraduate lab, what we measure is a smaller unit that is called the enzyme unit, U, which is the amount of enzyme which is required to either convert 1 micromole of the substrate per ml per minute, or 1 micromole of the product is formed per unit time. That is the enzyme unit, U. So now we come to our experiment that we are going to do, that is the amylase activity. To give you a background, we are going to study the activity of an enzyme that is called salivary amylase, which is found in your saliva, or pancreatic amylase, or alpha amylase. All these enzymes, I told you last time, are enzymes which are acting on carbohydrates, and they are responsible for the hydrolysis of the carbs. Okay, that is by the name amylase. Now, the salivary amylase or the alpha amylase, it acts on the 1,4-glycosidic linkages in the sugars, and it breaks them down. The most common substrate for salivary amylase is the starch. You have seen in the carbohydrate chapter that starch is actually a mixture of two polymers. One is amylose. This is 1,4-linked glucose. You can see the glycosidic linkage, the 1,4-glycosidic linkage here. This is the amylose, which is the linear molecule, and the amylopectin, which is the branched molecule, which has the main chain consisting of 1,4-glycosidic linkage and the branch chain consisting of the 1,6-glycosidic linkage. So our amylase or the alpha amylase or the salivary amylase, as I said, will cleave these glycosidic linkages and not these. Okay. So as I said, it acts on the 1,4 glycosidic linkages present in the starch alpha and not cellulose, because cellulose has beta 1,4 glycosidic linkage, and it will break down. It is actually a exoenzyme, so it will break down the starch into maltose units from one end progressively, leading to formation of, you know, 2-glucose linked by the 1,4 glycosidic linkage. That is maltose. Okay, so the starch is broken down into different maltose units. Now, how do we measure the activity of this enzyme? We understand if we take starch, we add amylase, react it with amylase, we will get maltose. But how to find out how much of starch has been converted into maltose? That I'm going to answer in the next slide. I would just like to tell you that this enzyme, the amylase, requires calcium ion for its stability. I will show the structure and tell you. And also it requires the chloride ion for its activity. Okay. This is the structure of your salivary amylase. This is your chloride ion in green, and this is the calcium ion. So the calcium ion is required for maintaining or for stabilizing the three-dimensional structure of the protein. If the calcium ion is removed from the amylase structure, you know, the three-dimensional protein structure opens up, and the active site is no longer available. So the enzyme loses its catalytic activity. Whereas the chloride ion, you know, it is required to accelerate the reaction. Okay. It is actually a cofactor for the enzyme, which is required for the acceleration of the enzyme catalyzed. Let us look at the mechanism of the enzyme catalyzed reaction. The mechanism is very, very simple. If you recollect, I'm just drawing the partial structure of starch, which is the rest of the glucose units here. I'm not drawing the OH groups here. You can do that yourself. Okay, so we assume that this is the rest of the glucose units here. Now, in the active site of the enzyme, you know, there are two aspartate units which are involved in the reaction. Okay. Now, your chloride ion, you know, is somewhere close to this aspartate group. Now, please understand, for the carboxylic group to ionize, the electrophilicity of this carbonyl carbon is very crucial. If the electrophilicity of this carbonyl carbon is high, you know, you have the lone pair delocalization taking place this way, and the proton is lost. So what this chloride ion does is that it reduces the electrophilicity of this carbonyl carbon, so that it is increasing the pKa of this aspartate. Okay. Now this proton is very, very important for the hydrolysis of the glycosidic linkage. Okay. See, please. Like this. That is how it is. Okay, so it is very, very important for the hydrolysis of the starch 1,4-glycosidic linkage. So if this proton is not available, you know, at the optimum pH of the enzyme, then the hydrolysis of the 1,4-glycosidic linkage will not take place. So what chloride ion does is it is increasing the pKa of this carboxylic group so that the proton is available, for the cleavage of the glycosidic linkage. This OR minus will take this proton and it will go as this is what you get. So this carboxylate group is involved in stabilizing the positive charge on the oxygen here. Okay. And in the next step, the water molecule will come. So the water molecule can come from below the plane or it can come from above the plane. Okay. So when it comes from above the plane, so you can complete the structure yourself there. You will get this type of arrangement, and when it comes below the plane, you will have this type of arrangement. So that is how the 1,4-glycosidic linkage in starch is cleaved, and the two aspartates in the active site of amylase, they help in the catalysis. This one is involved in stabilizing the positive charge on the oxygen, and this one is involved in donating the proton, which helps the cleavage of the 1,4-glycosidic linkage. Okay. So, we have added amylase to the starch solution, allowed the reaction to take place for some time. The amylase has broken the starch solution and it has given you molecules of maltose. But maltose is colorless, starch is colorless. How do you use spectrophotometry to identify how much of starch has been converted into maltose? So there comes the role of this molecule. This is called DNSA. Okay, 3,5-dinitrosalicylic acid. Okay, this 3,5-dinitrosalicylic acid, it will what it does is it is actually an oxidizing agent. So it oxidizes your maltose, which is a reducing sugar, to maltobionic acid. Recollect, in the theory class I have given you the structure of maltobionic acid, and it is itself reduced to 3-amino-5-nitrosalicylic acid. Okay, 3-amino-5-nitrosalicylic acid. Now this reaction takes place in strongly alkaline conditions, around 9 to 10 pH. Okay, so DNSA, you allow the reaction to proceed for some time, and then you add this DNSA. So what this DNSA will do is it will oxidize maltose to maltobionic acid and itself gets reduced to 3-amino-5-nitrosalicylic acid, which absorbs strongly at 540 nanometer. So the amount of 3-amino-5-nitrosalicylic acid will correspond to the amount of maltose which is produced, and the amount of maltose which is produced is the measure of the activity of amylase. Okay, that is the underlying theory in the measurement of amylase activity by the DNSA method. Okay. So I told you what is the role of chloride ion. It increases the pKa value for the aspartate group in the active site, so that the proton is available for the cleavage of the glycosidic linkage. And the role of DNSA, I told you that DNSA is acting number one as an oxidizing agent. So it is oxidizing maltose to maltobionic acid. And the other thing it does is that the 3-amino-5-nitrosalicylic acid is a colored species, absorbs strongly at 540 nanometers. So it is used to quantify the activity of amylase. And third, very important thing is that DNSA is made in strongly alkaline solution. That strongly alkaline solution immediately arrests the activity of salivary amylase. So DNSA solution is also used to bring the enzyme catalyzed reaction to a halt. Then the role of calcium ions. The calcium ion, I told you already, I have shown you the structure also. You have seen it is required for stability, the three-dimensional stability of the protein structure or the enzyme. If the calcium ion is displaced from the active site, the three-dimensional structure will open up and the active site will no longer be available for binding the substrate. So the enzyme will lose its catalytic activity in absence of the calcium ions. Then the role of Rochelle salt. DNSA preparation, while the DNSA reagent is prepared, some amount of Rochelle salt is added. This Rochelle salt, prevents the dissolution of oxygen in the reaction mixture by keeping the ionic concentration high. Now, if oxygen is dissolved in the reaction mixture, what will happen is it will introduce error by oxidizing the maltose to maltobionic acid. So you can understand that how it will affect or how it will reduce the amount of maltose which is available for reacting with DNSA, leading to an experimental error. Now let us look at the procedure for plotting the maltose standard curve. So like I have told you, in the Lowry's method, the measurement of the enzyme activity will consist of two steps. The first step is the plot of the maltose standard curve. So you plot an absorbance versus concentration curve for varying dilutions of maltose, so that you have a graph which you can manipulate, which you can extrapolate for an unknown solution of maltose and determine its concentration. Okay, so after having done that, we will do the enzyme activity. Okay, we will find out what is the absorbance of a solution where the starch has been acted upon by amylase to produce maltose. Okay, and by adding the DNSA from the standard curve, we will be, by extrapolation, we will be able to measure the concentration of the maltose in solution. And this concentration we will use to measure the enzyme activity. So you can understand that the measurement of the amylase activity will have two steps. As I said, first will be the plotting of the maltose standard curve, and second will be the measurement of the enzyme activity by using the maltose standard curve. So what you have to do is you have to take a set of 9 clean and dry test tubes, and you have to label them. First one, like we did in Lowry's method, first one will be labeled blank. The next 8 will be labeled T1 to T8. Okay, The next 8 will be labeled T1 to T8. To each of these 9 test tubes, you will be adding maltose as per the table given. I will show you the table like we did in the case of Lowry's method. I have listed the volume of maltose to be pipetted out, and then it has to be followed by addition of distilled water so that the total volume in the test tube is 1 ml. Okay. After that is done, you have to add 1 ml of DNSA. You have to cover the test tubes and you have to heat them in boiling water bath for 10 minutes. Okay? After boiling in the water bath for 10 minutes, you have to cool and you have to dilute with 4 ml water. Okay? And these diluted solution, you will use the blank to set your instrument to 0, and after doing that you will note the absorbance for T1 to T8, and using those absorbance values you will plot the A versus C curve. So what we are doing here is we are trying to measure the absorbance of different dilutions of maltose solution. You know how maltose is reacting with DNSA. Varying concentrations of maltose will give you different concentrations of DNSA, reduced 3-amino 5-nitrosalicylic acid, so which will give you different absorbance. We use that, as I said, to plot the maltose standard curve. Of course, I have told you last time also when we were discussing Lowry's method that after every addition, it is very important that you shake and mix the contents before keeping them back in the test tube stand. Okay. So that is how your table will look like. You can see that there are 9 test tubes here, the first one being the blank and the remaining is your T1 to T8. Here, the varying concentrations of maltose that you have to add, the stock solution is 1 mg per ml that we have prepared. So you can see if I am adding 0.2 ml and making the volume to 1 ml, I have diluted it 5 times so that it becomes 200 micrograms per ml. Likewise for others also I have done the calculation for the maltose in microgram per ml. And the remaining will be diluted with water. Shake it after adding water and then you add 1 ml of DNSA. Like when you start adding 1 ml of DNSA to your blank, you have to start your stopwatch. Continue addition of 1 ml to all the 9 test tubes and allow the test tubes to remain undisturbed for a total of 10 minutes starting here. Okay. After that, you boil them in the test tube for 10 minutes on a boiling water bath. Cool, dilute with 4 ml of distilled water and you measure the absorbance. Okay. That is, as I said, your maltose standard curve. Now, after you have drawn the maltose standard curve, you will take 2 clean and dry test tubes. Now, this is the enzyme activity part. We have done the standard curve, and this is the enzyme activity part, which comes now. So, we will label one clean and dry test tube as control, and the other clean and dry test tube as test. So in the control, you will first pipette out 1.4 ml of the buffer solution. Okay, that is the acetate buffer. We have given you the pH is 5.6, which is the optimum pH of the salivary amylase, the sample that we use here. We find that the activity of salivary amylase is maximum at this pH. 1.4 ml of the buffer solution you add, and then you add 0.1 ml of the given 1% sodium chloride solution. In the test sample, you have to add 1.2 ml of the buffer, 0.1 ml of the 1% sodium chloride solution, and 0.2 ml of the enzyme. See, in the control, there is no enzyme. So your control, as I said earlier, is enzyme-like. Okay? You incubate the mixture for about 5 minutes. Incubate the mixture for about 5 minutes. Both control as well as test. After 5 minutes incubation, you have to add 0.5 ml of starch to both control and test and shake. Okay. Then again, incubate for 20 minutes. And you must note down the temperature. You can do it at room temperature, but please measure the temperature because enzyme activity will vary with the temperature. Okay? So in our case, either we can do it at 37 degree or we can do it at room temperature, and we can measure the room temperature and we can say the activity of some library amylase was found to be this at this temperature. So that gives a more logical meaning to it. Then add 1 ml of DNSA to both the test tubes after 20 minutes. Please understand. The moment you start adding starch, here, you must start your stopwatch. 0.5 ml starch you add to control and 0.5 ml starch you add to test. Shake both. Your stopwatch is already on and you have to continue incubating for about 20 minutes. So when 19 minutes are over, you will be ready with your DNSA. Immediately as you finish 20 minutes, you have to add 1 ml of DNSA to both control and the test and shake them immediately. So the moment you add DNSA, your reaction will be arrested, and whatever maltose has formed, on heating, it will react with DNSA, will get oxidized to maltobionic acid and produce the measuring species, that is 3-amino-5-nitrosalicylic acid, which you measure spectrophotometer. So you have to cover both control and test with the foil and then boil on a water bath for 10 minutes. After the 10-minute boiling is done, cool, dilute with 3 ml water. Okay. This is the table that I was talking of. I have just explained it to you also earlier, how it is to be done. This is the control and this is the test. We have discussed this, how it is to be done. And then set the spectrophotometer to 0 with the control. Okay, so you are zeroing all other species except for your 3-amino-5-nitrosalicylic acid, which is formed by the reduction of DNSA by maltose. Okay, then record the absorbance of the test sample. Now, once you record the absorbance of the test sample, for example, it comes out to be this 0.5, you use your maltose standard curve, interpolate it, and you determine the concentration of your maltose in the solution in micrograms per ml, like this. Okay. And then after having found the concentration of maltose in micrograms per ml, you will calculate the activity of the enzyme formula, concentration in micrograms per ml into dilution factor, which is 50. What we have done is we have made a stock solution of 1 mg per ml and we have diluted it 1:50, to make the working solution which you are using. And when we calculate the activity, we are calculating the activity for the stock solution. So it has to be multiplied by 50. The total assay volume, the total volume of the solution that you are using for measuring the enzyme activity, that is 2 ml. Before you measure the enzyme activity, you can see here. Go back to the table here. Okay. That is your test sample. You can see if you add up the volume, it becomes 1.5 here and zero point 0.5 here. The total volume is 2 ml in which you are measuring the enzyme activity. Okay, you don't include this because this is added to arrest the reaction and to see how much of the product is formed here. Okay. Total assay volume, the enzyme volume that you added is 0.2 ml, the incubation time is 20 minutes, and the molar mass of maltose, that is 342 grams per ml. So you can get a shortened formula, that is your concentration in microgram per ml into 0.0731 micromoles per ml per minute. That will be the activity of enzyme. Do not forget to mention the temperature. So this is the activity of my enzyme sample at this temperature. Okay, so what precautions you have to take? Any enzyme assay, any biomolecular assay that you have to use clean and dry apparatus, because we have these experiments, these are very, very sensitive, and we are working with very dilute solutions. So a drop or two of water also can be, very detrimental in the overall result. Always use clean, dry, and labeled pipettes so that you do not mix up pipettes in, pipette should be used for pipetting out a particular solution also and not mixed with the other pipettes. Pipette very carefully so that you don't introduce any pipetting errors. Then shake the contents immediately on mixing. That is very, very important. And the incubation time and the heating time, is very, very important, because that is a measure of how much of the reaction has taken place and how much of your 3,5-dinitrosalicylic acid has been reduced to 3-amino-5-nitrosalicylic acid. So now we will do this experiment in the lab and see how the experiment comes out to be, and we will find out what is the activity of our sample.